one of my closest nieces has had a diagnosis of autism for the last 25 years, and to this day we still don't understand why she has autism, so that motivates me in part. I think genomics is really providing us a path for understanding why children develop developmental disabilities and autism more broadly. So by understanding the different types of events, things that can go wrong in the development of an individual, gives us insight in terms of the genes, and those shed light in terms of the pathways that hopefully we can actually develop therapies or cures for in the future. I think one of the things that I've realized over the last 25 years of work is that families with developmental disability often feel like they're alone. They don't feel like there's anybody else in the community that has exactly the same types of problems that they have. And one of the exciting things that has come to me, which is actually better than most of the research, that, the research is great, but is the ability to actually identify specific causes, specific mutations and specific genes, and then to unite people all around the world that have the same type of genetic cause. And what we have done is, by doing that, we've actually started to see that there are subtypes of intellectual disability, subtypes of autism. And it's been for me, a personal blessing actually to connect those families that feel so isolated, but now actually have found the 6th and the 7th family that has a child with the same type of disability. So it's not as if we're providing a cure yet. We're still, I think, quite a ways away from that. In fact, we don't even talk about cures in my lab. We talk about treatments down the future. But I think the first place to start is really by identifying the causes. Mm-hmm. And then once you identify the causes, then to bring those people together. Mm-hmm. And that is happening really at a rate that was previously unimaginable, even 10 years ago. We have been at the cutting edge of developing some of the new technologies that I think have revolutionized discovery of genetic causes for autism and developmental disability. And so I think, having that technology and then having access, to patient samples through clinicians has made this thing really a synergy that involves really 3 groups: the researchers pushing the technology, understanding the genetics, the clinicians who are at the front lines with the patients, and perhaps most importantly, the families, who contribute the samples, who contribute, they're very interested in staying in touch with us regarding this. And I think, to me, that's a dynamic trio, right, of investigator, clinician, and family. And when a child is born with developmental disability, the question is, how come this happened to us, right? Why are we alone on all this? And I think finding, even though we try to be really, open about this, that, we're not at the stage of cure for most of this. Families in general are so appreciative of that information, that it wasn't something that they did wrong in the 2nd trimester, right? It wasn't that extra glass of wine that they had, that it was, wasn't some other issue, right? That it in fact was this genetic cause. I think, to me, it's one of the kind of the cool things of doing genomics research, is being able to help people, and especially families that, often don't have a lot of help or resources. And one of the things that we've realized from, the work that we've done is that there are many different genetic causes that can lead to a child with autism or a child with developmental disability. And any individual cause maybe only explains a fraction of a % of all the cases. But when you start to screen tens of thousands or hundreds of thousands of patients, you start to identify the 10th and the 11th and the 12th case of a specific genetic cause. And then when you bring those families together, even though they're essentially all called, children with autism spectrum disorder or developmental disability, we start to identify subtle things that are actually more similar among those children with the same genetic cause than just children born with autism or developmental disability. And I think that's really important for 2 reasons. I think, number one, it makes families, realize that they're not alone, that they in fact have other families upon which they can actually form a support group and so on. And there's ways of handling, managing, or dealing with children with disabilities that, the advantage of having a 45-year-old mother talking to a 25-year-old mother are just huge. And a lot of clinicians would say, well, that's not actionable. But I disagree, because I think what you're doing is you're providing information which improves the quality of care for those kids. So I think I disagree. I think that's really valuable information. You also provide information for the family and say in terms of whether there's risk of having additional kids. So in many cases there isn't, right? So many kids actually are the result of a genetic mistake that has happened during the production of sperm or egg cells. And so we can say to them that there's no risk that if you have a second child, there's no risk than it would be for the general population. So I think that that's really valuable information for families. But the hope is, in the future is that by knowing the genetic cause, you know the gene that's affected, you know where that gene is expressed, you know when it's expressed, and you can actually start to think of treatments that may in fact improve the function of that gene that's been disabled or broken. And I think that's really powerful, because the idea that you would treat a child that actually has a gene that's shut off, that's usually expressed first week or two after birth, the same way as you would treat a gene that was expressed early in development, I think is a mistake. And right now, a lot of the treatments treat all kids with autism and developmental disability as if they're the same. And I think what we're advocating is that we need to start thinking about subtypes. And maybe whatever treatment works good for this particular child will not work so good for this one. And I think the way to sort that out is based on the genetic cause of disease.